Welcome to Biology 221. This is the first lecture. We're going to do an introduction. So to start us off, I'm showing you a beautiful picture. I want you to ask yourself if you recognize it. This is the Sandy River right here in Troutdale, so probably quarter of a mile, half a mile from school. That's the Troutdale Bridge. So a picture of biology that's close to where our campus is and really beautiful. And I'm showing you something like this to start us off because biology is the study of life. Something that I think is important to think about when we talk about biology is the difference between learning and studying. So I have you saying, tell your neighbor, because I want you to pretend that you're in class with other people while we do this. And as you're thinking about it, maybe you can tell your cat or your dog or your friend or your houseplant, but think about something that you are good at, and it doesn't have to be related to school. And then think about how you got good at that thing and how long did it take you to get good at that thing. For me, I love mushrooms. I studied botany as my undergrad, and I studied mycology for my graduate degree. And so I have spent years and years and years studying mushrooms, and I love them, but I'm not like the most amazing identifier of mushrooms. I studied taxonomy, and I'm not like immediately site ID for things. So I care about being good at identifying mushrooms, and I've practiced it continually. I read books, I go outside, I talk to my friends about it. That's how I learned how to do it, and it has taken me years. So I have two different mushrooms here I wanted to show you. The first one here, I found this on campus. This is a porcini. It's a beautiful edible mushroom. It's delicious, it's nutty, it's wonderful. And I also found these on campus. These are called Galerina. The common name for these are funeral bells. So about half of one of those caps would be enough to kill you. It's horribly poisonous, toxic. It would not be a good edible. And those two things don't look alike, but knowing the differences between them and their lookalikes is really important for identifying mushrooms. So I want you to take a moment, pause the video, and think about something that you're good at and how you got good at it and how long it took you to get good at it. The idea behind this is when you care about something, you end up studying it, but it doesn't feel like studying. When you try and learn something, you often practice it, you spend hours on it, you talk about it, you put effort into it, and all of those things are important for learning biology as well. So my goal is to try and get biology to feel like something that you are interested in, and so that when you study it, it doesn't feel tedious, but it feels like trying to learn something fun. One of the ways that we can actively try and learn something is with this technique called active learning. It's also called scientific design-based teaching. So we try and get students to think about ideas using their own knowledge as a barometer. It's called metacognition, when you think about what you know as you're trying to learn something. I have a couple article headlines I want to show you. This one here says, active learning and student-centered pedagogy improve student attitudes and performance in introductory biology. This one says, improved learning in large enrollment physics class. And I have these slides for you up on Blackboard so you can read the fine print later. Here's another article, an active learning strategy primer for achieving ability-based educational outcomes. And active learning increases student performance in science, engineering, and mathematics. Active learning means that when you are learning something, you're not just hearing it and then never thinking about it again. That's called passive learning. That's when you let something kind of wash over you. In active learning, I'm going to be asking you to think about the material. I'm going to have you draw it. I'm going to have you write it. I'm going to have you map it. I'm going to have you talk out loud, hopefully, And I'm going to set up the videos so that I'm talking to you and asking you to do these things, and I'll tell you to pause, give yourself time to do them, and then restart the video when you want to continue. Kind of like Blue's Clues, you know, when they talk to you on the TV and you're responding. So our first thing that we're going to do for active learning is think about the characteristics of life. I have an image for you. This is my cat. This is Parker. I want you to think about, how do you know that something is living and something else is not living? So how do you know that Parker is alive, but my pile of laundry is not? I want you to think of 5 characteristics off the top of your head. It turns out that when biologists classify life, we use 7 characteristics. These 7 characteristics of all living things are that living things are composed of cells, at least one or more cell. We call that the cell theory. Living things maintain an internal balance. That's called homeostasis. For example, we can't get too hot or too cold or we die. We can't get overhydrated or underhydrated or we die. We have to maintain the right amount of blood pressure or we die. That's homeostasis. The same thing is true for my cat, Parker. Parker has to maintain solutes and hormones and water, all of the other nutrients. He's maintaining an internal balance to keep himself alive. That would also be true for a single-celled organism. It has to maintain its internal environment, different than the external environment. The third characteristic of life is that all living things obtain and use energy. If you are a plant, you make your own energy using photosynthesis, and then you break the sugars that you made. If you are an animal, you have to eat food, and so you are going to consume food and turn it into energy. We'll talk about how your body does that later in the term. It's really cool. All living things can also reproduce. It doesn't mean they have to, but it means that they can. And even if an individual can't, like if a certain cat is sterile, like my cats have been fixed, they cannot reproduce, but their species can. So it's an on the whole ability to reproduce. So even though Parker will never have kittens, he is still a part of an organism group that is able to reproduce. So plants reproduce, bacteria reproduce, everybody has the ability to reproduce if it is alive. The next characteristic is that living things can grow and develop. Parker looks different now than he did when he was a kitten. And you look different now than you did when you were 1 year old. That's growing and developing. Even single-celled organisms will change over their lifespan. That's growing and developing. All living things respond to stimuli. It might be like a plant growing toward the sun, or if you've ever had a plant fall over and then it rights itself, it can feel gravity. That's called gravitropism. And so that is feeling the stimulus of gravity and responding to it. For us, we respond to smells, sights, tastes, sounds. That's all stimulus that we respond to. And living things can evolve. Evolution in biology means that things change over time, not individuals, but groups. So Parker does not look just like his parents. I don't look just like my parents. Every generation is a little bit different. And over time, species change. That's what evolution is. So if you cannot reproduce and cannot have differences in every generation, then you do not evolve, and we do not count that as a characteristic of life. So now that we've discussed the 7 characteristics of life, do you think that this is alive? What is this? This is the coronavirus. This image is from the CDC. It's called the coronavirus because these proteins are called spike proteins, and they make a crown around the outside. Corona means crown. So this coronavirus has all of these proteins on the outside, and then it has this gray membrane. Inside of it, there's a little bit of DNA or RNA. And the way that this virus works is that it will hook to your cell, push itself inside your cell, and it will trick your cell into reading its DNA and not your own. And so your cell starts to make more coronavirus instead of doing its usual job. So you will start to create more and more viruses that will exit the cell. It basically hijacks your cell and forces your cell to create more of it. This would be a really cool time to pause the video and look up how does coronavirus work, or how do viruses trick cells into making replicas of themselves. For us, I want you to think about those 7 characteristics of life. First, quiz yourself, can you remember them? And second, ask yourself, does this have that characteristic? Is this alive? The answer is maybe, right? Like, how many of the characteristics of life do you need to be considered alive? We know that a virus isn't technically a cell because it doesn't have any other components inside besides for that DNA I told you about. It does not maintain homeostasis. It doesn't have any like internal environment and blood pressure and like need to keep itself warm. It does reproduce, but it can only reproduce if it infects a host. It doesn't grow and develop. It doesn't use energy because it uses your cell's energy to reproduce. We don't think it responds to stimuli, but these little spike proteins do interact with your cell. So maybe you could count that. And then it does change. That's actually one of the reasons that viruses are so hard to treat, is because every time they infect your cell and your body starts reading their DNA, the DNA replication isn't exact, and so it makes changes with every generation. And change over time is our definition of evolution. But it also means that the virus that I am releasing into my body is different than the virus that I was infected with. So that's why flu vaccines have to change every year, because the virus itself changes over time. It's evolving. So do you think those 2 characteristics that got a check mark are enough to count this virus as alive? We can Google it. If we Google it, that's what I wrote in my Google bar. I got these 3 answers. Microbiologysociety.org, that seems pretty trustworthy. No, viruses are not alive. Okay. Carleton.edu, so that's a school. We generally trust schools. Are viruses alive? Viruses are definitely alive. So that's a yes. And then we have Ask a Biologist at ArizonaStateUniversity.edu. We trust Arizona State University. Are viruses alive? Some do not think viruses are considered alive. So like, what do you do when the answer is no, yes, maybe? How do you figure out what of these sources you want to trust? And to answer that question, when you look at Blackboard, there'll be 2 other videos under this. We have a video for the evaluating sources using the CRAP test, CRAP with 2 A's. And we have a video about peer review. How did you feel? This is my other cat, Max. If you have any questions, email me. Email me anytime. I love emails. They go right to my phone. I'm happy to answer them. Please email me here instead of using Blackboard. It'll make me respond much more quickly. And I hope you had a fun first lecture. I will talk to you soon. Bye.